Okay, dear students, you have already learned how to do functional group tests for aldehyde, ketones, and carboxylic acids to analyze compounds systematically. Today, we are going to discuss how you will identify compounds which have carbohydrate and esters. Uh, Let us start by first doing the tests for carbohydrates. The group test for carbohydrates is the Molish test. So let us see how the test is first done, and then we will try and understand the chemistry of the test. So you will take in a test tube 0.1 gram of the organic compound, okay? And you will add 2 ml of water to this, meaning you have to prepare a very dilute aqueous solution of the organic compound. To that, you will add 2 to 3 drops of Molish reagent, which is a solution of alpha naphthol in ethanol. And then you will add along the side of the test tube concentrated sulfuric acid, 1 to 2 ml. Allow the content to stand. What you will observe is you will observe a violet colored ring at the junction of the two layers. If you observe this, then you conclude that your compound is a carbohydrate. Okay, let us see visually how the test looks. This is how you will get a violet colored ring, which will be coming at the junction of the two layers. In between the two layers, this is the bottom layer, will be sulfuric acid, and this is the aqueous solution at the top. And this is the violet colored ring that you get here. Okay. Let us now understand the chemistry of this test. The chemistry of this test. What happens is if you have any simple sugar, for example, glucose, concentrated sulfuric acid being a dehydrating agent, will convert it into this compound. This is 5-hydroxymethyl furfuryl. Okay, this is 5-hydroxymethyl furfuryl, which will then condense with 2 molecules of alpha naphthol in the acidic medium to give you these violet colored species. This is the species which is formed by the condensation reaction of 2 units of alpha naphthol with 1 unit of 5-hydroxymethyl furfuryl. And that is how you will get this violet coloration at the junction of the two layers. This is the aqueous layer. This is your concentrated sulfuric acid at the bottom, which appears some greenish in color. Okay, so that is your group test for carbohydrate. If the group test is positive, you will go on to do the other special tests for carbohydrates. The first of those is the Barford reagent test. The Barford reagent test is actually a test which is used to distinguish monosaccharides from disaccharides. A Barford reagent is a 7% solution of copper acetate in 1% acetic acid. So basically, you have cupric ions in the acidic medium. The test is very similar to the Fehling solution test that you do for the aliphatic aldehydes. What you have to do here is that you have to take 0.1 gram of the organic compound, add 2 ml of water, and then you add 1 ml of a freshly prepared Barford reagent. Heat the mixture in a boiling water bath for about 5 minutes. If you get a brick red colored precipitate settling at the bottom of a blue colored supernatant liquid, the inference that you draw is that the compound is a monosaccharide, can be glucose, fructose, mannose. What happens is this, as I said, is a test which distinguishes the monosaccharides from the disaccharides. The monosaccharides, being more reactive than the disaccharides, are oxidized by cupric ions in the acidic medium. Now, please understand that in failing solution, you are using cupric ions in alkaline medium, which is a stronger oxidizing agent. So this will react with both monosaccharides as well as disaccharides. But Barford reagents has cupric ions in the acidic medium, which is a weaker oxidizing agent. So it only oxidizes the more reactive monosaccharides. Okay, let us see the color chemistry first, and then we will see the reaction. See, this is the sugar solution. This you have added the Barford reagent. And then when you heat, at the bottom of the blue colored solution, you will get that red colored precipitate of cuprous oxide being formed. Okay. Let us now understand the chemistry of this test. See, this is glucose. For example, if you have glucose in your molecule, then it reacts with cupric ions with water in mildly acidic solution. It is oxidized to corresponding carboxylic acid, that is gluconic acid, and the cupric ions are reduced to the red colored cuprous oxide, which is giving you that red precipitate. You can see that red colored precipitate at the bottom of the solution. So that confirms the presence of a monosaccharide. If you do not get any red colored precipitate in the Barford reagent test, then the conclusion is that your compound is a disaccharide. Okay. The next is the test to distinguish between a reducing and a non-reducing sugar. Now, the reducing sugars will reduce Tollens reagent to metallic silver, which will give you the silver mirror, whereas the non-reducing sugars will not do that. Okay, so how to do that test? Let us see. You will take 0.1 gram of the compound, add 2 ml of water, and then add 2 ml of freshly prepared Tollens reagent. Heat the test tube in a boiling water bath for about 5 minutes. So what will happen is you will get a silver mirror being formed at the inner walls of the test tube, which will confirm that the compound is a reducing sugar. It can be glucose, fructose, mannose, lactose, maltose, the disaccharides. Okay. Let us see the chemistry and the color chemistry of the test. So you see that how to prepare Tollens reagent, you should understand first. What you will do is you will take about 1 ml of silver nitrate solution, add a drop or two of 10% sodium hydroxide solution, which is available in the lab. You will get a brown, black colored precipitate of silver oxide or silver hydroxide being formed. To this solution, dropwise, you add aqueous ammonia until the brown-black colored precipitate just dissolves. Okay, so the resulting solution will be a Tollens reagent. So you add 2 ml of this solution to the aqueous solution of your organic compound. And when you heat it on the water bath, this will be the silver mirror, metallic silver being deposited on the inner walls of the test tube. So that is the Tollens test. Now, after you have got the silver mirror test, it is important to decompose the metallic silver. So you immediately add dilute nitric acid so that silver dissolves through the formation of silver nitrate and it is run down the running water in the drain. Otherwise, silver, if left exposed to air, it forms a highly explosive compound that is called silver fulminate. Okay, so that is why you should never leave the metallic silver unattended after you have performed the silver mirror test. Let us see the chemistry of the test now. If you have glucose, on reaction with a Tollens reagent, that is ammonical silver nitrate solution, glucose will be oxidized to the corresponding gluconic acid salt, and the silver ions will be reduced to metallic silver, which is giving you that silver mirror. So that will be the chemistry of the Tollens test. Another test that you can do to distinguish between a reducing and a non-reducing sugar is the Fehling solution test. So you can either do the tolerance test or you can do the Fehling solution test in the laboratory. You will take 0.1 gram of your organic compound again and dissolve it in 2 ml of water. To this, you will add a mixture of 1 ml each of Fehling A and Fehling B. That means you first prepare a Fehling solution by mixing equal volumes of Fehling A and Fehling B, and then add 2 ml of that solution to the aqueous solution of your organic compound, and then heat it on a water bath, boiling water bath for about 5 minutes. A brick red colored precipitate of cuprous oxide, if you get, again confirms that you have a reducing sugar. If the test is negative, then you have a non-reducing sugar. So the test is very similar to the Barfoed reagent test I told you a while ago. You will get a red colored precipitate being formed. It settles at the bottom of the supernatant blue colored solution, due to the formation of cuprous oxide. Let us see the chemistry of the Fehling solution test. Now, if you know what is Fehling A, Fehling A is aqueous solution of copper sulfate, and Fehling B is alkaline solution of Rochelle salt, that is sodium potassium tartrate. When you mix equal volumes of Fehling A and Fehling B, you get copper tartrate being formed. That is copper tartrate, which is deep blue in color. Okay. So this then will react with your sugar. For example, if you have glucose, and you heat it with your solution of Fehling A and Fehling B mixture having copper tartrate. So basically it has copper 2 plus ion in the alkaline medium. So again, in the alkaline medium, you will get the gluconic acid salt being formed, and the cupric ions are reduced to the red colored cuprous oxide. Now, the important question that you have to understand here is, when in the Fehling test, you require only cupric ions in the alkaline medium, why do you have to take the trouble of keeping Fehling A, making Fehling A and Fehling B, and then mixing the two solutions together and then doing the Fehling test? Why can't you prepare an alkaline solution of copper sulfate and do the test? The answer is very simple. The copper sulfate, if you dissolve it in alkali, it precipitates as copper hydroxide. So the cupric ions are no longer available in solution for performing the test. That is why copper sulfate and the alkali, they are kept separately. When you mix them together, cupric ions are held in this complex by the Rochelle salt. And when you heat with the aqueous solution of sugar, in the boiling water bath, slowly, cupric ions are released from this complex and they react with sugar to oxidize it, and themselves they get reduced to cuprous ions, giving you that red precipitate. I hope you understand the chemistry now. Next is the rapid furfural test. Rapid furfural test is actually a special test for those sugars which are existing in the furanose form. So it can actually be used to distinguish between glucose and fructose. You know that fructose exists in the furanose form, where as glucose in the free form exists mainly in the pyranose form. So how you do this test is, you will take 0.1 gram of the organic compound, add 2 ml of water, and then add 1% alcoholic solution of alpha naphthol and 6 to 8 ml of conc HCl, and you boil the contents on the flame. When you heat it, you will get a violet coloration in the solution, which confirms that you have sugar having a furanose form like fructose or sucrose. Now, if you notice, this test is very similar to the Molish test that we have discussed as the general test for carbohydrates. The only difference here is that concentrated sulfuric acid in the Molish test here is replaced by conc HCl, which is a comparatively weaker acid than conc sulfuric acid. So what happens here is the sugars which are already in the furanose form, like the fructose and the sucrose, can easily form the 5-hydroxymethyl furfuran, which is required to give you that violet colored complex, even in slightly weaker acid, HCl, as compared to concentrated sulfuric acid. And then they will go on to react with 2 molecules of alpha naphthol to give you that violet colored solution that we have seen already in case of Molish test. So I hope you understand. If the test is negative, then you are looking for a compound like glucose, which is existing in the pyranose form. Okay. Now, let us see what derivative you can prepare for carbohydrate. Once you have confirmed that you have a carbohydrate present in the organic compound, normally for carbohydrates, the preferred derivative is osazone. Let us see how this derivative is prepared, and then we will see the chemistry of this preparation. So you will take 0.5 grams of the sugar, add 3 to 4 ml of water, and then 1 gram of phenyl hydrazine hydrochloride and 1.5 grams of sodium acetate. So the ratio of the sugar is 1 to 2 parts of phenyl hydrazine hydrochloride to 3 parts of sodium acetate in a boiling tube. Add a glass rod. Cover the reaction mixture, the neck of the boiling tube is covered with a cotton plug. And then you clamp it vertically in a boiling water bath. Gradually, you will observe that the solution will acquire a yellow coloration because of the formation of the osazone. So you allow the mixture to heat for about 15 to 30 minutes. Filter the product, wash it well with water, dry, and take the melting point. Normally, osazones are pure and they do not require recrystallization. While filtering, what you can do is you can wash the osazone precipitate with very dilute HCl, and then you wash it with water. What it does is the washing with dilute HCl will remove any unreacted phenyl hydrazine from your osazone precipitate, and so it will be practically pure to take the melting point. Let us understand the chemistry of this test. What happens is if you have, say, glucose, and you react it with excess of phenyl hydrazine, it forms glucosazone You will study in theory that the product formed in case of glucose and the fructose is the same. That is explained by something called the Amadori rearrangement, which you will do in details in your theory class. Okay. So this is glucosazone. Now let us go on to the preparation of ozone for non-reducing sugars like sucrose. Now, sucrose is a non-reducing sugar. It does not have a free carbonyl group, so it cannot react directly with phenylhydrazine hydrochloride. So what is done is that sucrose is first hydrolyzed to a mixture of glucose and fructose, and then it is reacted with phenylhydrazine hydrochloride in sodium acetate to prepare the ozone. Mind you, sucrose is a disaccharide. When it is subjected to hydrolysis in the acidic medium, it forms one molecule of glucose and one molecule of fructose. And I told you in the previous slide that both glucose and fructose, when they are reacted with excess of phenylhydrazine, okay, they will give you the same product. You can call it glucosazone or fructosazone. The product is the same. In fact, all the compounds, C2 epimeric compounds, which are differing from each other in their arrangement on C1 and C2 carbon, all those sugars will give you the same ozone. Okay. This can be explained by Amadori rearrangement, as I said a while ago. So how to do the hydrolysis? You will take 1 gram of sucrose, add conc HCl 2 to 3 drops in 10 ml of water, and transfer this acidified solution to the test tube containing your sucrose. Transfer the content to a round bottom flask, a 50 ml round bottom flask. Add 1 to 2 pumice stones and you reflux the contents using air condenser for about 30 minutes. After that, you cool the contents, allow the contents to come back to room temperature. Transfer them to a conical flask and then you concentrate the content, boil the contents to about 2 ml. To this 2 ml solution now, you add phenylhydrazine hydrochloride, 2 grams, and sodium acetate, 3 grams. Again, loosely cork the flask and heat it on a boiling water bath for about 15 to 30 minutes until you get the yellow precipitate of ozone being formed. Again, filter the precipitate, wash it first with very dilute HCl. You have dilute HCl solution in your laboratory. You can dilute it 3 to 4 times and wash it with very dilute HCl and then wash it with water and then dry. And then you can take the melting point of the resulting glucosazone or fructosazone to confirm the presence of sucrose in your molecule. So this is the chemistry of carbohydrates, how to do the tests and how to prepare the derivative of carbohydrate. This is how the crystals of ozone appear. Next, we will go to the functional group test for esters. Now, the group test for esters is the hydrolysis test. You will take 0.5 gram of the organic compound or 1 to 2 drops, dissolve it in about 1 to 2 ml of ethanol. Okay. Then add 1 drop of 1% sodium hydroxide solution and then 1 to 2 drops of phenolphthalein. If the pink color is discharged in cold, you can add another 1 or 2 drops till you get a permanent pink color in the solution. Do not add sodium hydroxide in excess. Now, warm the contents on the boiling water bath for about 5 minutes. If you observe that the pink color disappears or fades, it is confirmed that your compound may be an ester. Let us understand the chemistry of this test. you have an ester, say, let us say ethyl benzoate, and I am hydrolyzing this in alkali, okay? You will get benzoic acid and ethyl alcohol. Now, if you have added optimum amount of NaOH, on hydrolysis, the benzoic acid that is formed will drop the pH of the solution. So the pink color of the phenolphthalein will disappear. If, however, you have added sodium hydroxide in excess, then the faint amount of benzoic acid that is formed in the solution will not be able to drop the pH of the solution enough to discharge the pink color of phenolphthalein. That is why I said that you have to be very, very careful when you are adding sodium hydroxide. Do not add sodium hydroxide in excess. Okay. The next is the confirmatory test for the esters, that is the hydroxamic acid test. For that, you will take 1 to 2 drops of your organic compound in a Keene test tube, add 0.2 grams of hydroxylamine hydrochloride, and 5 ml of 10% sodium hydroxide solution. You gently boil the contents of the test tube on a Bunsen burner for about 1 to 2 minutes. Cool the solution, acidify it with dilute HCl. See that the blue litmus is turning red. Okay, and then cool, and then you add few drops of ferric chloride solution. If you get any violet or wine color or magenta colored solution, on addition of ferric chloride, then the ester is confirmed. Now let us see the chemistry of the test. So let us again take ethyl benzoate, okay, and we have added hydroxylamine hydrochloride and sodium acetate. Let us first understand what will happen when hydroxylamine hydrochloride and sodium acetate react together. See, this is a HCl salt of hydroxylamine. So when you add sodium acetate to this, what will happen is you will get the free base hydroxylamine being formed, which will combine with ethyl acetate, and you will get hydroxamic acid being formed. That is the hydroxamic acid. And of course, you will get ethyl alcohol. Now, this hydroxamic acid will combine with ferric chloride, and it will give you ferric hydroxamate. That will be your violet or wine-colored precipitate being formed. Okay, so that test. Now it is important that you do a blank test here. Some phenols will also give you violet, blue, or colored solution with ferric chloride. If that is the case, then this test is not useful for those compounds. So do a blank test. Repeat the same procedure, everything except for the fact that you do not add hydroxylamine hydrochloride during the while performing this test. Okay, so if you are getting a violet coloration or a wine color or a magenta colored solution, then do not perform the hydroxamic acid test for these esters. Another very important thing here is the hydroxamic acid test is actually a test for presence of acyl group. So beside esters, this test will also be given by amides. It will also be given by anhydrides, and it will also be given by acid chlorides. So it is important that These compounds, which can be identified by other tests, are ruled out when you confirm the presence of the ester in the given compound. Let us see what derivative you can prepare for esters. So for esters, the simple hydrolysis product is used as the derivative. So for doing the hydrolysis in the large scale, what you should do is you should take a round bottom flask, take 1 ml of the organic compound in that, add 15 ml of 10% sodium hydroxide solution. So whenever you are doing the derivative preparation, the reagents, should not be the bench reagents that you use. Either you ask the lab staff to prepare 10% sodium hydroxide solution and use it, or you can prepare your own 10% sodium hydroxide solution and use it. Okay, add 1% one or two pumice stones to the RB flask, and then reflux the contents using an air condenser on a sand bath for about 50 minutes. Check for the completion of the reaction. Cool. Acidify the contents by addition of dilute sulfuric acid. Check if the blue litmus has turned red. Cool. And filter the contents. Wash with minimum amount of cold water because the carboxylic acid which is formed may be soluble in water. Dry. Recrystallize that from hot water or hot aqueous alcohol depending upon the solubility of the compound, and take the melting point to confirm your compound. Okay, so with this, I hope you have understood how you are going to do the tests for the carbohydrate as well as for the esters. Thank you.